everybody, I'm Tall.
And I'm Small.
And today we are doing Khan Academy's MCAT Prep Biomolecules DNA Technology questions. Okay, the first question is, plasmid G is cut with the restriction enzymes EchoRI, BAM-HL, and HIND ILL. There's like 3 lines in a row. It could be anything, I guess.
Dr. Christensen would hit you right now because those, so RI is actually, an I, but the BAM-H1 and HIND3, those are Roman numerals.
Well, I blame Khan Academy for not making that more apparent. So, so the plasmid G is cut with the restriction enzymes, those, in various combinations. The sizes of the resulting fragments are seen in the table below, and you guys will just have to, read the table. Which of the following is an accurate representation of plasmid G? So they give us 4 answer choices, and unfortunately, I cannot show you guys these answer choices over audio. So when we look at the table, the chart, it says that BAM cuts at 14 kilobases and 20. BAM plus Echo cuts at 3, 5, 12, and 14. Echo plus HIND cuts at 5, 13, and 16. BAM plus Echo plus HIND cuts at 3, 4, 5, 10, and 12. Okay, so how do we even approach this?
Do you know?
No, I have no idea.
Okay, so I have an idea.
Okay.
And hopefully I'm not totally off base here. So to me, reading this chart means that these 3 together would produce these cuts, and so 3, 4, and 5 would have successive cuts right next to each other. So we would have like a 1 kilobase distance between each cut there, and then 10, and then 12, and so those would be also close to each other with a 2 kilobase distance between them. And so for me, the one that looks like this might be...
Okay, so this one has Echo, BAM, and Echo right next to each other. And it says that Echo, BAM, and HIND cut at 3, 4, and 5? I'm like beyond... This is so beyond me. I have no idea. So
if you're looking at a circular plasmid, right? At some point on this plasmid, there's a first kilobase section.
Okay, so I just tried to answer, and it wasn't correct because I literally have no idea.
Yeah, I don't know what I'm
there's
doing.
no way we can convey this over audio.
Okay.
So I'm running through these answers right now to try to figure out which one it is. It's the first one. I just want to look at their hints because maybe their hints will say something. So plasmids are circular pieces of DNA. So 1 cut will make 1 fragment. 2 cuts will make 2 fragments, etc. All right, next hint. When EchoRI and BAM-HI are used together, the 14 kilobase fragment created by BAM alone is preserved. This implies that both Echo sites are within the same BAM-bound fragment. Okay. When HIND and Echo are used together, a 5 kilobase fragment is preserved. And the only way the restriction enzymes will produce the fragment sizes listed in the tables if 2 Echo sites are bound by 2 BAM sites while a HIND site is opposite them. Huh.
Oh my goodness. It literally tells us the sizes of the resulting fragments. Wow. Okay, so 3 kilobases, 4, 5. Oh my goodness.
Yeah, so there's not really much we can do over an audio medium with this. However, the only advice I could give is draw out a circle and then start to draw exactly the kilobase sizes that they give you in the table. Draw those between the actual cuts and see what makes sense. Okay, next question. In nature, restriction enzymes are meant to protect viruses from bacteria, bacteria from other bacteria, humans from bacteria, or bacteria from viruses. Okay, so I kind of think I remember this from my micro class or my genetics class, that restriction enzymes, while we use them, we utilize them in the lab, but they're produced or they're originated from bacteria to protect them from bacteriophages.
Yep, exactly. And so that's how we name them too, right? So the E core 1, 2, however many there are from E. coli. And so
Okay.
that's how we chop them up and name them.
Okay. And sure enough, that was the answer. Bacteria from viruses. Okay, next question. BAM H1 recognizes 6 base pairs of DNA in a palindromic DNA sequence. If the first part of the sequence is given below, what is the rest of it? Okay, so the thing you want to notice here is that, okay, so they give us a 5' to 3' end, and it says CCA, and it shows you the point where BAM cuts. The only thing you need to know here is the fact that it said 6 base pairs, and it said palindrome.
Exactly.
So palindrome means that when you get halfway through reading it, you start reading what you just read in reverse.
Yep. It's the same forwards as it is backwards.
It's like a mirror image. So if you had CAA, which is what we have here, then the backwards of that is AAC. But I could see them easily like messing with us with the 5' to 3' stuff.
Exactly. And that's something I want to point out. By convention, we write all nucleic acids from 5' to 3'. And so sometimes when they throw in answer choices, you'll immediately see that AAC. But because we've written it from 5' to 3', that's the opposite. You know, that's just that first segment again, and not that mirror image that you mentioned for palindrome.
Well, you mean CAA.
Oh, yes. I'm sorry. Yeah.
Yes. So it should be from 5' to 3' AAC.
Yeah.
Because it would be continuing that same strand.
Oh,
Okay.
we gotta stop doing this at night.
All right. So it would be AAC. And no, that's not correct. Oh my gosh. What would it be? Oh, wait. No. It's got to be the palindromic sequence. Is it the opposite of each? So it would be TTG? It is.
Oh, no.
Let's look at those hints. Okay, so the answer was 5' to 3' TTG. Okay, so the complementary strand to the one given must start with 3' GGT, 3 more, and then 5'. A palindromic sequence of DNA will read the same sequence 5' to 3' on either strand. Remember, a palindromic sequence of DNA is slightly different from a palindromic number, such as 12321, or words such as stats. The final part of the sequence recognized by BAM is 5' to 3' TTG in order for it to be palindromic. Huh, so does this make sense to you?
No.
It doesn't totally make sense to me either, other than the fact that if you put TTG after CAA, then when you read the complementary strand, it will be CAA-TTG.
That's right. And that would give us the sticky ends of cutting both with a 2 nucleotide overhang on both sides.
Okay. So that's it. You have to think about the complementary strand as well while you're doing this.
Yeah.
Okay, next question. A researcher has identified the location of the gene that codes for protein X and has managed to excise it from the DNA. She has also generated cDNA that codes for the protein. If she runs these two sequences through gel electrophoresis, what should she expect to see? Okay, and they give us a couple of different pictures showing different kinds of gels that have either that have both cDNA and the excised DNA on them. So reading back through the question, they managed to excise the gene that codes for this protein, and then also generated cDNA from it. So
the only thing I know about cDNA is that it's a library.
Right, but for our purposes here, we're looking at size.
Right. So I would think that would be a lot bigger.
If it's cDNA.
But I don't know. I really don't know anything about it. That's the only thing. And it's from a memorized biology class some point in the distant past. Right.
So I'm thinking they just have to be the same. Because if it's the same gene, so let's just say we have the actual gene in front of us. If we chop it out, and let's just say it's 100 base pairs, and then we also make a cDNA copy of it, wouldn't that also be 100 base pairs? I think.
I would hope so.
Yeah, so I think they would have to run the same, and there's one choice that does show them both running the same distance. So it's wrong. All right. So tell me, Con, what is the correct answer? Because none of these seem to be making sense. Okay, it is one where the cDNA ran further than the excised DNA. Let's find out why. Excised DNA would be the original sequence, while cDNA is the reverse engineered from the protein. Gel electrophoresis. Oh, introns. The excised DNA would still have introns. If you reverse engineered it basically from the protein.
Okay, yeah.
Okay, so we would expect the cDNA to be smaller, therefore it ran further than the excised DNA.
Yeah,
Dang.
that's something good to point out. The human genome has a crazy amount of so-called junk DNA that we think might actually affect a lot of maybe epigenetic things that we're not really sure of yet, but
right.
Yeah, most of those are introns.
Okay, so if you had excised DNA, there's going to be introns in it. If you had cDNA, you're copying it from mRNA, right?
Yes, you're kind of reverse doing things, right?
Yeah. Okay, next question. A student was attempting to make cDNA that would code for protein Y. They began with an isolated and amplified sample of the target post-transcriptional mRNA, and all the tools required to complete the task. However, at the end of the experiment, the student was left with a molecule containing ribonucleotides. What most likely went wrong? Ribonucleotides. So are they talking about ribosomal RNA, you think?
Yeah.
Okay, so
well, I think ribonucleotides is just a form of RNA. So maybe they didn't get all of the U's out.
Instead of deoxyribonucleotides.
Right.
They're talking about ribonucleotides. Oh, right, so there might be some U. Okay, so they want to make cDNA for this one protein. They have mRNA, and they have all the tools they need. But when they're done, they got a molecule of supposed cDNA that had ribonucleotides in it. So what went wrong? Answer choices are: the student forgot to add the DNA polymerase, the student added RNA polymerase instead of DNA polymerase, the cloning vector already transcribed the DNA, or the student forgot to add reverse transcriptase. I'm leaning towards the student added RNA polymerase instead of DNA polymerase. Because if you have a template RNA strand, no, RNA polymerase only makes RNA from DNA, right?
Yeah.
Ah.
Yeah, and so DNA polymerase, while it can use an RNA primer, I don't think can use an RNA template.
But the one thing that can use an RNA template is reverse transcriptase.
Exactly.
Okay, so I'm going to go with that. And it's right. Awesome. Thanks for the help, Small. Okay, a student adds Tack polymerase nucleotides and a primer to a test tube. What procedure are they most likely going to do next?
All right, dry lab person.
Okay, dry lab person's going to read the answer possibilities and try to squeeze the answer out of Small. Southern blot, gel electrophoresis, microarray hybridization assay, or polymerase chain reaction. Okay, so it's obviously not Southern blot.
He's gauging my face for reactions.
Gel electrophoresis. You kidding me? It wouldn't be gel electrophoresis.
Still looking at my face.
Microarray hybridization assay. I actually know what that one means.
Yeah, you bioinformatics person.
Absolutely. Polymerase chain reaction. I actually think it's polymerase chain reaction because I know PCR uses primers.
That is a good bet. Also because it says Tack polymerase.
That might be a given. I had no idea.
Yeah. Do you know what Tack polymerase is?
I don't. Teach me.
So we use a polymerase from thermophile microbes, so bacteria that can handle heat and actually prefer to grow in heat. That's their optimal conditions. And we use their polymerase because it won't denature at such a high temperature, whereas if we attempted to use something like human polymerase, it wouldn't work at that high temperature that we need to use in PCR.
That's really clever. And sure enough, that was the answer. Polymerase chain reaction.
I would know the difference between Southern blot, Northern blot, and Western blot. There's no Eastern.
Yes.
Southern would be DNA. Northern would be RNA. And Western we also use all the time on proteins.
Very cool. Okay, next question. PCR is an excellent method of generating copies of target DNA. If a single piece of double-stranded DNA is put into a PCR machine, how many double-stranded DNA segments will there be after 3 rounds? Okay, so we have 1. That makes 2 after 1 round. And then 2 would make 4 after 2 rounds. And 4 would make 8 after 3 rounds.
Okay, hold on 1 second.
We have a single piece of double-stranded DNA, goes into the machine. Yeah, because I think it reads, I remember seeing somewhere it works kind of like semi-conservative replication, where you have the original strand and then, the original like 2 strands kind of wound up together, and then you separate them, and then you add nucleotides to one of the original strands, and then those separate, you add nucleotides to those strands. Therefore, I think 8 would emerge from that.
Yes, and I just needed to do the math in my head. Your brain works quicker than mine does.
Okay, so we have 2 options: 8 segments with the 2 original strands paired, or 8 segments with the 2 original strands on different segments. I think I kind of gave that away when I said it was semi-conservative replication.
Yep, that's a good point. And also, one of the first things we do is to, I don't know if they call it denaturing, but we take apart the strands, so
yeah,
they would both act as templates for the next round.
Mm-hmm. Oh yeah, and then when it cools back down, which is called annealing.
Yep, at like 72 degrees, I think, maybe.
When it cools back down, you now have those original strands with a new strand attached to it, so semi-conservative replication. And
Yep.
that was right. Okay, next question. A scientist is trying to express gene X in bacteria. After cloning the cDNA from gene X into a plasmid with the appropriate cDNA and transfecting the plasmid into bacteria, the scientist noticed that he forgot to add antibiotics. What will greet him the next day? Is it a petri dish with separate colonies of transformed and untransformed bacteria, a mostly normal petri dish, antibiotics are just used as a safeguard against other bacteria, a petri dish completely overrun with untransformed and transformed bacteria, or a petri dish with only untransformed bacteria? The antibiotics are required for transfection. Okay, that one I think is right out. Antibiotics are definitely not required for transfection.
Right. Knowing that transfection means using a viral delivery method.
Right. Let's look at the first answer was a petri dish with separate colonies of transformed and untransformed bacteria. Separate colonies. I don't think that one's correct.
So I think the reason that it's incorrect is because when you're using a plasmid that you have designed, you would typically need to have an ORI site, so with some restriction enzyme cutting sites so that you can introduce the gene you want to introduce. And then you need to have some selectable marker like antibiotic resistance so that the bacteria that grow are only the ones that successfully were transfected.
Okay, so we can definitely kick out a mostly normal petri dish. Antibiotics are just used as a safeguard against other bacteria. And a petri dish completely overrun with untransformed and transformed bacteria. I feel like that's the answer. You just wouldn't be able to select from them for the correct ones.
Yep, you lose your ability to select when there's no antibiotics present. Good.
However, no, I'm gonna not second guess myself and go with my gut, and it was correct. Very cool. Okay, next question. Which procedure is least effective for detecting a specific DNA sequence in a sample? Southern blot, polymerase chain reaction, gel electrophoresis, or microarray? All right, so we are trying to find a specific DNA sequence in a sample. Southern blot is DNA, right?
Mm-hmm.
Okay, polymerase chain reaction. Polymerase chain reaction, if you had the primer for your DNA, then you could just throw it in there and see if it starts to amplify your sequence. Gel electrophoresis, that is talking about, that would pull DNA based on its size.
Mm-hmm.
So if you cut it up, so maybe you could have like some kind of endonuclease activity that could cut up your specific DNA and hope that there's no others that are the exact same size. Microarray is looking at quantities.
It's expression, right? Though, so it's RNA, I think.
Oh my gosh, you're right. Oh, you're totally right. It's looking at RNA expression. That's what I was thinking. I was like quantities. Oh no, it's wrong. Is it gel electrophoresis? I should have gone with my gut. No, I probably would have picked microarray as well. Okay, let's look at the hints. So polymerase chain reaction uses specifically constructed primers to amplify a specific DNA sequence. Thus, you can tell if the target sequence was in the sample based on the... Okay, yeah, we knew that. A southern blot is one of the most efficient methods for detecting the presence of a specific DNA sequence in a sample. It works via hybridization of the target sequence to a probe. If the probe latches on, the target is there. Okay. Gel electrophoresis separates fragments based on size and is not nearly as specific as the other techniques mentioned above. Maybe I forgot what microarray. Well, I've read microarray data before. I would hope that. But maybe if you had expression of it, But I mean, I think gel electrophoresis, I could easily see why it really would not work. Okay, so let's move on to the next question.
Oh, and I do want to correct myself real quick. I think I remember saying Ore site, which is just the origin of replication. And I think I meant to say multiple cloning site, which is the area with all of those restriction enzyme cutting sites. So
Okay.
Forgive me.
Okay, so the next question, last question. A DNA sample was sent off for Sanger sequencing. I know what that means. The results are shown below. What is the sequence of the template strand? Okay, so they show us a square representing a gel or a piece of paper, and it shows from negative to positive. It shows A, C, G, and T, and it gives us a bunch of lines. So there's 2 lines under A, 2 under C, 2 under G, and 3 under T. However, I want to ask you, do you notice anything about these lines that might be very useful?
I don't know specifically what you're leading me to, but I was just going based off, I know that Sanger adds like a dideoxy nucleotide and it ends the chain early.
Yes.
And so then we could separate them based on size in a gel.
Mm-hmm.
And then I would decide which one was the first and which one was last based on not amino versus carboxy, but 5' versus 3'.
Right. So that the one running the longest would be the shortest. So let's just say that we knew that we terminated it at the very first nucleic acid. Well, if we look at our square, the one that ran the longest was an adenine. Nice. So that means that that's probably the very first one. Then the next longest run was a cytosine, followed by thymine, then guanine. So if we read this graph, which was kind of what I was alluding to, if we read it in the order that it gives us, it would be A, C, T, G. Do we have that down here anywhere from 5' and 3'? A, C, T, G. Yeah, we have it in 2 places. Okay, so A, C, T, G, and then T, C, A, G, T. T, C, A, G, T. We have that for answer choice D. Very last question. And it's not right. No. Okay. Oh, no. Oh, I think I know what the problem was.
What was the problem?
Isn't this doing the complementary to the actual strand? I think this is doing the complementary to the actual strand. Therefore, we need to do literally what we just did, but in reverse, which would be... it
asked us what the sequence of the template strand was.
The sequence of the template strand.
So you're right. So it was the opposite of what we ended up picking.
Dang. Read carefully, people, but strong work.